Good morning to everyone. My name is Naina, and today I'm going to give insights about ADMET properties and how the molecule comes out from the lab bench to the life-saving drug. So let's get started. So ADMET is a foundation of pharmacokinetics and pharmacodynamics. So pharmacokinetics is what drugs does to the body, and then pharmacodynamics what body does to the drugs. So process, it starts from the administration of drug. It pharmacokinetic and dynamic shows overall journey of the molecule or the compound, how it interacts with the body, what happens to it once it get metabolized, and what happens. So what is ADMET? ADMET stands for the absorption, distribution, metabolism, excretion, and toxicity. These are the key aspects and key parameters of pharmacokinetics and pharmacodynamics. And these are important because they play a very major role in therapeutic activities or toxicities. So let's get into it, how the process occurs. So first, when we administer the drug, first step is administration. So there are several routes of administration, including intravenous, subcutaneous, transdermal, oral. Oral is very common, and inhalation. So depending on the route of administration, the drugs goes into body and gets absorbed into the bloodstream. Second is distribution. So once the drug is administered, it gets absorbed. Once it is absorbed into the bloodstream, it gets distributed throughout the body. Like it goes to the different tissues and organs throughout the body, and through the bloodstream. So bloodstream is a highway which passes or which is a mediator to travel the drug from site of administration to the site of action. After distribution of drug, what happens? Drugs get distributed throughout the body and it goes to the liver. In liver, what happens? There are several enzymes like a family of cytochromes P450. So those what they does, they metabolizes the drug and break down the drug. So after breaking down and metabolism, the drugs goes to the kidneys and it get excreted from the body. So importance, why we need ADMET screening? Any idea? Okay, so we need ADMET. Why? Because as yesterday Dr. Parshuraman sir told that ADMET, because of ADMET, we will get to know many things about drug like pharmacokinetics and pharmacodynamics. And around 39% of the drugs are failing because of these pharmacokinetic properties in the last stage or the uh, clinical trials, etc. So to minimize that or to identify such failures, we have to do such screening before only in the first step. Then also toxicity plays major role. Sometimes what happens, the drug is there, it is giving good action, like it is a good therapeutic agent, but it is having very toxic properties, like it is toxic in nature. Sometimes it shows like hepatotoxicity, skin sensitivity, irritation. So what happens? It again fails. So it won't carry out. So for that, we need to carry out the ADMET properties before the studies. So we'll get into the absorption. What is absorption? Absorption is the process by which the drugs moves from the site of administration to the bloodstream, into the bloodstream. So because as we know, it has different, like we have different type of route of administration. So different route of administration gives the different pathways. So if the oral, like if we are taking in the oral, so if the drug is administered orally, what happens? It gets into the stomach and it get absorbed by the lining of stomach or the intestine. And after that, it goes to the bloodstream. If the drug we are taking, which is topical, if you are using any topical creams or ointments, what happens? The drug get absorbed through the skin or the mucus layer, it passes through the skin or mucus layer and it goes into the bloodstream. And third, sometimes what happens, the drugs travels from the cell membranes. So there are two pathways, passive transfer and active transfer. So in passive transfer, what happens? Drugs goes inside the cell without the use of ATP, that is energy. So that is passive. Here we don't need energy to travel the drug. But in active transport, what happens? A drug needs ATP to enter the cell. So these are the pathways. Then while describing about the absorption, one thing got into my mind, that is bioavailability. So what is bioavailability? Bioavailability is the proportion of drug which which enters into the systemic circulation in its active form. Active form means without metabolizing, and it shows the therapeutic effect. So it is expressed as in the percentage. And see, because if we take IV, so IV intravenous, what will happen? It will directly goes into the bloodstream. That's why it has 100% bioavailability. But if we are taking drug which is orally, so orally, what happens? The drugs goes into the stomach, it gets metabolized there, then it goes to the liver, there it gets metabolized by enzymes. And so before reaching to the systemic circulation, it gets metabolized. So the bioavailability is less. So this is a pathway, and that terminology is called as first pass metabolism. So what are the key factors affecting absorption? So first is physicochemical properties. In that, solubility plays an important role, because to get absorbed, it should be soluble. Second is lipophilicity. To pass any barrier, it should be more lipophilic. Third, ionization, molecular size, etc. Second is route of administration. As I already explained, route of administration plays important role. If we are taking IV, it will 100%, it will gets into the bloodstream. Then formulation. Formulation plays important role, because there are, with active ingredients, there are several excipients, and even the particle size of formulation matters, like nanoparticles for blood crossing the blood-brain barrier we use, like that. And last is biological and physiological factors. It includes GI, gastrointestinal pH, then transit time, enzymatic activities, blood flow, etc. And there are some methods too, like by which we in vitro techniques we can evaluate these absorption parameters. Those are these pampa and this one. Then drug distribution. So what is the drug distribution? Now we understood what is absorption. It is absorbed, the drug is absorbed into the body. But after that, what will happen? It will go and distribute to the different type of uh, various type of tissues and organs inside the body. So it is very important. Why? Because this is when it travels, then only it will reach to the site of action, and it will show therapeutic or toxic effects. So what are the key factors which are affecting the distribution? First is plasma protein binding. So what is plasma protein binding? In plasma, there are some proteins like albumin. So what happens is, when the drug is passing through from the bloodstream, there are the proteins. Drug will go and bind to the plasma protein. So it will not go and bind to its site of action. It will go and bind to the plasma protein. So ultimately, what happens? It will reduce the therapeutic effect. Second is volume of distribution. Third is blood-brain barrier. In blood-brain barrier, I think if you all know, it won't cross all the compounds. There is some criteria, like compound should be lipophilic in nature, smaller in size, then it will pass. And also blood placental barrier. Blood placental barrier is what, when the pregnant woman is there, and at that uh, in the pregnant woman, if we are taking any drugs, so the drug should not cross to the fetus. This barrier is playing important role in that. So some drugs like alcohol, those will pass. So that's why they tell us not to consume during the pregnancy. So what is volume of distribution? Volume of distribution is a main pharmacokinetic parameter that represents the theoretical volume in which the drug would need to be distributed to achieve the same concentration in the plasma. So it provides an indication of how extensively a drug distributes into the body tissues versus remaining in the plasma. So it is calculated by this formula: dose administration by IV by plasma concentration. And if the high volume of distribution indicates extensive distribution into the tissues, and low suggests that the drug is primarily confined to the plasma. So distribution. So how distribution process occurs. So as I told, route of administration plays important role. If we are taking from from IV injection, it will directly goes to the heart. If we are taking orally, it will come into the stomach, then intestine, stomach, then it goes to the liver. Here it will get metabolized, and then it will go and pass. So like this, and if we are taking something like inhalation, if somebody is there with asthma, so the doctor will give them some pump, or from the some nasal drops. So if we are taking some nasal pump or something, it will directly goes to the lungs and acts. So that's how the distribution occurs. Then drug metabolism. So drug got administered, then absorbed, distributed. Then what about metabolism? Then what happens is metabolism occurs in the liver. So there are certain enzymes like cytochromes which will help in drug metabolism. So there are two phases. First is functionalization phase, and second is conjugational phase. So in first phase, what happens? These like reaction, there are certain reaction in which they modifies the drug molecule in such a certain way that molecule should get it's molecule should slightly, it should become more water soluble, and it will provide substrate for phase 2 reaction. So these are the certain enzymes, and there are example like conversion of products like codeine to morphine via this cytochrome 2d6, like that. These are the examples of cytochromes which are mainly important for the metabolism of drugs. Then second, conjugational phase. In conjugational phase, what happens? The drug, or like the drug which drug are not metabolized by phase 1, those drugs, or which are metabolized, those drugs, those metabolites, those are conjugated to the some molecule and making them more hydrophilic. So if it is more hydrophilic in nature, it will go directly to kidney, and it will easily gets out from the urine, gets eliminated from the urine. That is the main purpose of it. And there are some reactions like glucuronidation, in that conjugation with glucuronic acid, then acetylation, methylation, etc. So once the drug is metabolized, what happens? It goes to several, like it goes to kidney. Some drugs are excreted from the liver, then some are excreted from the sweat, saliva, even the breast milk and lung. And mainly more drugs are excreted from the kidneys. And toxicity. So toxicity also plays a very important role, because if the pharmacokinetics properties are good, but if the drug is toxic, if the like action is good but drug is toxic, then it is of no use. It will again go and fail. That is why it is very important. So it is very important to check the hypersensitivity or the hepatotoxicity or carcinogenicity of the drug before the any trials. Then, so now we will go into the demo, how to check the ADMET properties. So there are several tools, freewares and a paid tool to check the ADMET properties. So we are going with the freewares today. So first, I'll show the demo with the pkCSM. So you can access this website pkCSM, and you will get this window. After getting here, you have to click on the pkCSM here. Then after that, you will get like this some window. So how you will do it? Here you need some smiles to upload. So you know how to create the smiles. If your drug is already FDA approved or already it is there, then you can go to the PubChem. You can put your name of your drug, and you will get something like this. So you can click here, and you down, if you scroll, you will get a smile. I have taken example of paracetamol because it is like widely used world over. So you can take this smile, you can copy it, and you can go and paste it over here. Like you can upload a whole file also if you have like many compounds, not one compound. If you have more than one compound, you can upload some notepad file here, or if you have one compound, you can directly put smile there in this column. And if you have don't you, if you don't have smile, you can generate through open babel or chemra also if you have designed that compound. So after putting your smile there, you have to scroll down and you will get like this window. So you can predict any property. You can predict either absorption, distribution, metabolism, excretion, toxicity, one by one, or like if you want togetherly, you can directly click on ADMET. Okay, once you click there, you will get like this result. You have here molecular properties. In that you are getting molecular weight, log P, rotatable bonds, hydrogen bond donors, acceptors, and surface area. And that side you are getting all the ADMET properties, like absorption first. In that, water solubility is there, the intestinal absorption, skin permeability, etc. So this is the review of one by one. And how to interpret this. So like whatever the value you have got, like suppose for water solubility, whatever the value you have got, this is the standard range for the water solubility. So what we have got is minus 1.0661. So that is showing it like it is low soluble, like the solubility is less. Then like that same intestinal absorption, if you can see, you have 91% of intestinal absorption. That means the drug is getting absorbed into the, like 90% it will get absorbed in intestine. Then skin permeability and substrates of glycoproteins, etc. Then this is the distribution properties. You have bound, then blood-brain barrier, if it is crossing the blood-brain barrier or not, CNS permeability, etc. And these are the metabolism parameters. In that, you can see all like even the substrates and inhibitors are saying no. That means it will metabolize easily. The drug is not substrate for this cytochrome 2d6, and it is not inhibitor of any cytochrome. Then excretion and toxicity. So like this, we have the ranges. According to that, you can interpret the results. Thank you. Now we'll get into the demo. So you can directly go to the pkCSM. You can type pkCSM. You will get first website that you have to click. And as I told you, you'll get this window. So you can go and click over here pkCSM. Okay. Once you click, you will get this window. So now you have to upload your smile. So how you will upload smile? First, you have to create smile. So you can go to PubChem or something. So you have gone to PubChem now. You can type any drug, like whichever you want, you can type. You can suggest any drug name. Anything. Ciprofloxacin. See, you will get it. So you got like this. Click here. Scroll down. You got the smile. Copy it and go to the pkCSM website. You can paste it here. Okay. After that, what property you want? You want all the properties? So we'll go for the ADMET. See, you can see here it is running. So it will take little time and you will get here structure also, structure of the drug. You can see one by one the results are coming you got for water solubility. It will run till that time. I'll show one more like the another one, another website which is Swish ADME. You can access this also from this website. I think the website is under review. Yeah. So here also same thing. You can either draw your structure or you can either put your smile. So we have already smile now. So I'll just copy and paste it and run. If you click run, down you can see molecule 1. You have got all the your properties for this molecule. And if you want to download this, you have here retrieve data. You have here one option of CSV. You can click on that. You will get it downloaded in the Excel sheet. Now we'll go to pkCSM. It is still running. So which is fast? Swish ADME. In Swish ADME, you can see the even the drug likeness. You have like different rules, including Lipinski rule, Ghosh rule, and all. So it will show if it is showing any violation or it is obeying the rule or not. In ADMET, you will get only the, I mean, in pkCSM, you will get only the results. Here you will get some different parameters also. So I'll suggest you can use both. It is still running. Okay. Any questions? Yes.
After running, we get different type of parameters. So in a research paper, what we have to publish, what factors we need to consider for publications?
Okay. Like you can generally give what are very important. Like you can give like if it is crossing the blood-brain barrier, as I told. Uh, for C and even the CNS permeability you can give. Then lipophilicity, molecular weight, like that, and hydrogen bond donor and acceptor, that also you can give, because that will play, like you will get to know no how many like hydrogen bonds donor and acceptors are there. So it will impact on your like molecular dynamics or docking, because you will get to see there some interactions also with hydrogen bond. You can add that, and even you can mention about the toxicity also, because it will also affect. And like I told, it is showing the excretion total clearance also. So like whatever the major things, if you feel you can tell to the publication, like you can mention in that, you can upload.
What are the major parameters?
Major, so toxicity and excretion you can mention. Metabolism, actually we usually mention all the parameters, but like it's on the your choice, I mean.
Can you show CCDME?
Yeah.
What is TPSA? Physicochemical parameters.
This one,
Yeah, TPSA.
that is topological surface area.
Any important?
Here in pkCSM, you have got the surface area, no? Like how much surface area it will cover.
Any importance in?
It has some like range. So, minute. Like importance of surface area. Uh,
Yes, please, can you repeat the question? Thanks, mate. Surface area. So usually surface area will be playing a major role where this property, again, when we are discussing with the MD simulations, right, there also it will be playing a role. What happens is like the surface plays how compatible it is, the drug structure. Because like this is the thing in the sense, what is the surface area, what the distance, how far it is there. And moreover, based on that, what do we call, the chain length will be there, like the linking. Example, CH3 bond CH3 means single bond. That will be more flexible. In case CH2 double bond CH2, based on that saturation and unsaturation point of view, this surface area will vary. Why? Because I can stretch and I can fold. So that stretchability will be reduced. So this, we don't have any exact, what do we call, numbers, or this is the range it should be present. Not like that. For that particular structure, what is the surface area user? So how it will be playing a major role in the sense, when the drug binds to the protein, so that will affect in the surface area, because SASA will be saying that we can't able to do in a Desmond. That SASA will be performed using GROMAC software, solvent accessible surface area. When the drug binds, no, slowly the protein structure will start to shrink. Shrink in the sense that more affinity will be created. So for that purpose, it will be calculated. So this property will be taken over there. Any other questions, queries? Any online questions? No online questions. No, that's fine. You can tell.
Any queries to be clarified? Thank you to the session speaker for sharing knowledge about ADMET studies. Before we start with next session, I request participant to sign in attendance sheet.